Hello, good day. So my name is Mukhtar Mohammed Ches, matric number 186456. So I'm about to present my final year project. So my title is Thermophilic Biohydrogen Production using attached biofilm on activated carbon from food waste. So my supervisor is Dr. Shakina. So for the introduction, currently the main source of energy are fossil fuels due to its depletion and the negative effects it has on environment has led to an increased interest of hydrogen as an alternative source. Production of hydrogen through anaerobic dark fermentation is eco-friendly way and was found as a viable alternative source to replace fossil fuels. Food waste and food processing waste, which are abundant in nature and rich in carbon content, can be a great source for sustainable biohydrogen production. So the feasibility of hydrogen production from food waste was being investigated. So I'm proposing a renewable way of getting energy from hydrogen. So this way is anaerobic dark fermentation. Anaerobic means without oxygen, and dark means without light. So this method is environmentally friendly, and then food waste is problematic. So we kind of find a solution for it. So my objectives that is the acclimatization of the hemophilic attached biofilm on granular activated carbon using synthetic medium. The determination of and the second objective is the determination of hydrogen production from attached biofilm and different concentrations of chicken, rice, and bread. So for my first objectives, uh, I will about to acclimatize for 14 days to activate the microorganisms using GAC with synthetic medium, and then later on I will determine the effect of different sugar concentrations of chicken, rice, and bread, and which one is the best that can give us the highest hydrogen production. So for the materials and methods, we divided into three parts. So for the preparation part, inoculum and synthetic medium preparation, food waste medium preparation, biofilm development through cell acclimatization. And for the analysis part, sugar concentration, which is total carbohydrates, TC, and TS and TSS. So, and monitoring biohydrogen production, gas chromatography. So, and one more thing is total volatile suspended solids and the experimental setup. So during preparation, an inoculum was obtained from oil mellifluent (bit) and preheated at 80 degrees Celsius for 60 minutes to inhibit the growth of methanogenic population. Granular activated carbon made from coconut shell was purchased and synthetic medium prepared containing a mixture of glucose and xylose as a source of carbon and other components. Food waste were blended with distilled water into three different concentrations, which are 10, 30, and 50. The biofilm was developed through cell acclimatization for 14 days in a modified squadron bottle bioreactor in a water bath shaker at 100 rpm and 60 degrees Celsius, and subcultured after 24-hour cycle. So subculture means the synthetic medium was changed half of the working volume. For example, if the working volume was 70, we changed the 35 of the synthetic medium, which is half. And then a 2 days experiment was followed by where inoculum containing synthetic medium with microorganisms put into the food waste. So 2 days experiment was conducted using inoculum that we get from this part, and then analysis of liquid and gaseous samples were taken, which was included analysis of sugar concentration, TC, by using phenyl sulfuric method, and TS and VSS, an analysis of biohydrogen gas using gas chromatography GC. So this is the schematic diagram of water displacement method to measure the volume of hydrogen produced. So this is the Squadron modified Squadron bioreactor. Here is a water bath shaker, and this there is a distilled water here, and this is the container for collection of gas. This cylindrical container, and this container contains acidic solution of pH 2. I mean, it's deionized water with acid of pH of 2, HCl. So here is the water bath shaker, and here is the container, and this is the cylindrical for water gas displacement method. So this is the 14 days for acclimatization of thermophilic attached biofilm on granular activated carbon using synthetic medium. So this is the GAC, and here is the synthetic medium. So it's being the synthetic medium is feeding the microorganisms that's attached to the biofilm on the GAC. So this is the food waste samples in thermophilic water bath shaker with syringe for collection of biogas during the experiment. So the food waste is in there with the inoculum, and this is for gas collection. For the results and discussion, during discussion, development of immobilized cells on GAC for stable biohydrogen production was investigated. The HPR obtained was highest at 2.163, and the stable hydrogen production reached an average of 72 hydrogen gas with HPR of 1.678. The effect of the concentration of food waste in biohydrogen production was found out that it can be obtained high amount of hydrogen yield in high concentrations of food waste. The 50% concentration of food waste contained chicken, rice, bread, and they produced 55 ml, 33 and 52 ml of biogas respectively, which is equivalent to 2.988, 1.173 and 2.825. So according to the effect of the type of food waste in biohydrogen production, chicken has produced the highest amount of biogas. So this is during acclimatization, the volume of biohydrogen production, and the numbers of days. So it's been conducted for 16 days, and then the synthetic medium with the microorganisms was removed and put it into the food waste at this date. So at the beginning, the biogas produced was fluctuating, and then when the days go by, it become almost constant. This is the HPR produced with the versus number of days. So this is the sugar concentrations. So total carbohydrates available in different types and concentrations of food waste. So this is before the experiment, and this stuff after the experiment. So as we can see from the graph, we can see that the total carbohydrates available in the food waste is decreasing. This is due to the microorganisms feed the carbohydrates that are in the food waste. So this is the concentrations of food waste when it's 50%, and the amount of hydrogen gas they produced. So this is for 80%, and this is for 10%. And the effect of type of food waste in biohydrogen production. So chicken was found out to be produced the highest biogas, which was 55 ml. And this is the GC results. As we can see from here, this 77.52 hydrogen was produced. So the objectives were achieved successfully. And then here is the comparisons in the previous studies on the hydrogen production from various types of food waste. So this 77% in biogas. So for conclusion and recommendation, it can be concluded that with high concentrations of food waste, this is a great substrate for stable biohydrogen production, especially those are rich in carbon. And for recommendation for better food waste digestion, the experiment period should be increased to monitor further on the soluble metabolites formation of hydrogen production.